Hello, my dear students. I once again welcome you to my class. Today, I will be taking a class of Kingdom Fungi. 11th class, Unit 1, Chapter 2, there, Section 3. As we have discussed earlier, we know well that R.H. Whitaker classified all the living organisms into 5 kingdoms. What are they? Monera, Protista, Plantae, Animalia, Fungi. In earlier classes, we tried to understand the characteristics of the Monera Protista. In today's class, let us try to understand the characteristics of the kingdom Fungi. The study of fungi is known as mycology. For a long time, even today, the fungi are studied in botany subject. For a long time, people thought that the fungi are plants, but in the course of evolution, they have lost the chlorophyll. They have become heterotrophic, became either saprophytes, parasites, or symbionts. So that's why they have been studied in botany. But Whitaker proved that the fungi are quite different from the plants with respect to particularly cell wall, with respect to nutrition, with respect to reproduction. They are quite different. Fungi constitute unique kingdom with many specialized characteristics. These specialized characteristics differentiate the fungi from the plants. Exhibit a great variation in morphology and habitat. A great variation in morphology. There are unicellular forms, multicellular forms, filamentous forms, microscopic, macroscopic like mushrooms, puffballs. They live in different habitats. They live in soil, they live in water, fresh water, marine water. They are parasitic on plants, they are parasitic in human beings, animals. Ecologically, they play a great role, decomposers. So, thus, there is a great variation in morphology and habit. Fungal body is made up of slender thread-like filaments called hyphae. Thread-like filaments. They are called hyphae, remember. Hyphal network is called mycelium. All the hyphal threads form a network-like structure that is called mycelium. Remember these two words, hyphae, mycelium. Filaments are called hyphae. The network of filaments, network of hyphae is called mycelium. Hyphae without cross septa and multinucleate are called cenocytic hyphae, just like in Mucor, Rhizopus, Pythium, Phytophthora. The hyphae are present, but without cross walls. It is a tube-like structure, multinucleate. Such hyphae are called cenocytic hyphae. Hyphae may be septate. As we go to higher fungi like Ascomycetes, Basidiomycetes, they become septate. That means hyphae are divided into cells because of the formation of the septa, dividing into cells. Cell walls are composed of chitin. This is a distinctive feature of fungi. In plants, the cell wall is made up of cellulose, but here it is made up of chitin. This chitin is also present in arthropods. And again, there is a variation. Within the chitin, there is a variation. It is also called fungus cellulose and poly. Cells are uninucleate. If the hyphae are divided into cells, those cells may be uninucleate in some species. Some species it may be binucleate in some hyphae, or these binucleate are called a dikaryon. Di means two karyon nucleus, or multinucleate. For example, in Deuteromycetes, Ascomycetes, the hyphal cells possess many nuclei and they are genetically different heterokaryon, they are called. Nutrition, they are heterotrophic because they don't have the chlorophyll. They lost the chlorophyll or they did not develop the chlorophyll from the beginning. Anyhow, they are achlorophyllous without chlorophyll, therefore nutritionally they are heterotrophic. Absorptive type of nutrition, they absorb the nutrients through the body surface, through the hyphal surface. Nutrition wise, the fungi may be saprophytes, grow on dead materials, absorb the nutrients. Those which grow on dead, non-living, organic materials, they are called saprophytes. They may be parasites, plant parasites, animal parasites, human parasites, or simply sometimes obligate parasites, total parasites. Symbionts, they make symbiotic, mutually beneficial association with the plants, especially with plants. Mycorrhizae, myco, fungus, rhiza, root. A symbiotic association between the fungus and root of a higher plant is called mycorrhiza. I repeat it. A mutually beneficial or symbiotic association between a fungus and the root of a higher plant is called mycorrhiza. The mycorrhiza is quite common in angiosperms. This is also found in gymnosperms. The fungus helps the plant root in turn helps the fungus, especially the phosphorus type of nutrition. Different types of mycorrhizae are there. Then lichens, we will study them in details. A symbiotic association between a fungus and alga. A symbiotic association between a fungus and alga is called lichen. So these are different modes of nutrition in fungi: saprophytes, parasites, symbionts, mycorrhizae, lichen. Again, there is a great variation. Sometimes the saprophytes may become parasites depending upon the condition, or parasites may become saprophytes. If the living materials are not available, living plants are not available, they go for saprophytic mode of nutrition. Even here also sometimes it is true. Then reproduction, fungi reproduce by vegetative fragmentation. The filaments broke down into small fragments. Each fragment, if it falls on a suitable substratum, then it develops into a mycelium. Chlamydospores, resistant spores. Under unfavorable conditions, the hyphal cells form thick-walled spores. They are called chlamydospores. Arthrospores, the hyphal cells automatically break into small cell fragments. They are called arthrospores. Asexual reproduction by juice spores, motile juice spores in lower fungi like Phycomycetes, juice sporangia. Inside the juice sporangia, juice spores are produced. The juice spores may be uniflagellate, biflagellate, and under moist conditions, if a film of water is available, they swim and go to a different place, germinate and give rise to the mycelium. Or conidia, dry spores produced on conidiophore. This is very common in Deuteromycetes. Sexual methods, oospores, in Phytophthora, in Pythium, oospores are produced as a result of sexual reproduction. Oogonium, Antheridium fuse and form a diploid oospore. Or zygospore in Zygomycetes like Mucor rhizopus. Isogametangia fuse and form zygote. Or ascospores. In Ascomycetes, inside the ascus, 8 ascospores are produced. Basidiospores, in basidiospores, upon the basidium, 4 basidiospores are produced. Sexual reproduction stages, 3 stages. A true sexual reproduction: plasmogamy, karyogamy, meiosis. Again, plasmogamy, karyogamy, meiosis. What is plasmogamy? 2 gametes, male gametes, female gametes, they should come together, unite. That union is called plasmogamy. 2 protoplasms. Once the 2 protoplasms unite, then male and female nuclei fuse with each other and form a diploid zygote, that is called karyogamy, union of male and female nuclei. There is a difference here. Plasmogamy, union of the protoplasms of male and female gametes. Still the nuclei are maintained differently. Here, union of the 2 nuclei forming a diploid nucleus, zygotic nucleus. Then at one or other stage, the diploid zygote undergoes meiosis division, reduction division, forming haploid spores. So in a true sexual reproduction, there are 3 phases: plasmogamy, karyogamy, meiosis. There is another mode of reproduction that is called parasexual reproduction. These 3 stages will be there, but not in a regular sequence. Dikaryotic phase. Di means 2, as I told you, karyos nuclei. If a cell consists of 2 nuclei, that is called dikaryotic phase. Results if there is a time delay between plasmogamy and karyogamy. In plasmogamy, as I told you, the 2 protoplasms unite. Protoplasm is single, but 2 nuclei. That is called dikaryotic state. Suppose if there is a delay between the plasmogamy and karyogamy, a dikaryotic phase results. Sometimes this dikaryotic phase may become dominant, as in Basidiomycetes. It is a dominant phase in Basidiomycetes. Fungi classification. This is very old classification. After this, several classifications have been proposed. But for easy understanding, we have to adopt, we have to take up one or other classification. This is a very easy class. For beginners, it is a convenient classification. Phycomycetes, pronounce it Phycomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes, Phycomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes. Now let us try to understand the characteristics of these. Phycomycetes grow in aquatic moist places. Phyco, water. Saprophytes or obligate parasites on plants like Phytophthora, like Albugo, Peronospora. They are obligate parasites, total parasites. They don't have the saprophytic phase. Mycelium aseptate, cynocytic. Another word, aseptate or cynocytic. Cynocytic, another word, cells or the tube-like structure, multinucleate. Without septa, multinucleate. Sexual reproduction, flagellated zoospores. The name itself indicates zoospores, zoo animal. Spores, animal-like spores, move with the flagella. Or non-motile aplanospores like mucor, rhizopus. The spores are produced in sporangia, but they are dry, they are minute, light, blown away by wind. They are called aplano, non-motile. Spores produced in sac-like sporangia. For zoosporangia, zoosporangium. For aplanospores, simple sporangium. Sexual reproduction, gametangial contact or gametangial copulation. Contact, simply they come together. Copulation means they unite with each other. Contact like this. Copulation here. Fusion of gametes. Fusion of similar gametes, isogamous. Isosimilar. The male and female gametes or gametangia or similar. Isogametes, isogametangia. Fusion of dissimilar gametes, anisogamous or oogamous. The difference between these two, anisogamous means male will be smaller, female will be somewhat larger. But here in oogamous, the male gamete is small, the female gamete is egg. Female gametangium is known as oogonium, very large. Sexual spores, oospores or zygospores. Then examples, rhizopus, commonly known as bread mold, mucor, albugo, white rust fungus. It causes disease on mustard. Then Phytophthora, an important fungus. Lead blight of potato. Rhizopus, mucor, albugo, Phytophthora. So this is albugo, cystopus, earlier it was called. Now these are the host cells. These are conidiophores, sporangiophores. The sporangia or spores are produced in basipetal succession, ruptured epidermis. This is typical rhizopus, rhizoids, mycelium, stolon. This is called stolon. The sporangiophores, stalk-like structures. Sporangia, inside the sporangia, spores. This is the broken sporangium, the spores are being released. This is rhizopus. Mucor will be similar to this except the rhizoids. Major difference. Ascomycetes, commonly known as sac fungi. The ascogonium will be sac-like structure. Therefore, they are known as commonly sac fungi. Ascogonium, a sac-like structure. They may be saprophytes, decomposers, parasites, plant parasites, coprophilous, The fungi that grow in dung, on the surface of the dung, coprophilous. Mycelium branched, multicellular septate. Yeasts are unicellular. Yeasts belong to Ascomycetes. These are considered as primitive Ascomycetes. Unicellular sometimes form pseudomycelium. Asexual reproduction by conidia produced exogenously on conidiophores. Sexual spores, ascospores, produced inside sac-like ascus that I will show you. Inside a sac-like structure, club-shaped structure. Fruit bodies, ascocarps. As a result of sexual reproduction, they produce the fruit bodies. In general, these are called ascocarps. Again, in ascocarps, different types. Clistothecium, perithecium, apothecium, like that. Clistothecium, perithecium, apothecium. Examples: Aspergillus, black mold, economically important, produces citric acid. Penicillium, blue mold, very important fungus. Penicillium chrysogenum produces penicillin antibiotic, a very popular, famous antibiotic discovered by Alexander Fleming. Claviceps, ergot fungus on rye plants, produces ergot toxin. The disease is ergotism. Neurospora crassa, it is commonly red mold. This has been extensively used in genetic studies. One gene, one enzyme theory was proposed by Beadle and Tatum while working on Neurospora crassa. Commonly known as morels, truffles. These are edible and very delicious. Here you can see this is a Penicillium conidiophore phylloids or sterigmata. The conidia are produced in a chain, basipetal chain, oldest, youngest. Then this is ascus, as I was telling you, this is ascus, sac-like structure. Inside this ascus, always 8 ascospores. Ascospores are always 8 sexually resultant spores. These asci, ascus singular, asci plural number, produced inside the ascocarps. This is clistothecium, this is perithecium, this is apothecium, this is another type of perithecium. Clistothecium, completely closed. Flask-shaped structure, perithecium. Cup or saucer-shaped structure, apothecium phasiza. Basidiomycetes. Common forms of basidiomycetes are rust fungi, cause rust diseases. Smut fungi, smut diseases. Mushrooms, puffballs, bracket fungi. These are the common names for basidiomycetes. Mostly saprophytes, wood decomposers, but some, just like Puccinia, Ustilago, Tilicia, they are parasites. Mycelium branched septate. Vegetative reproduction by fragmentation. The hyphae break into small fragments, pieces. Asexual reproduction generally absent. It is common in lower fungi, lower organisms. Sex organs absent. No well differentiated sex organs. The vegetative cells function as the sex organs. A copulation takes place between the vegetative cells, that is called somatogamy. Somato here, vegetative cells. Union of vegetative cells is somatogamy. Again, union between the two vegetative cells is known as somatic. Dikaryotic phase present, a dominant phase. Sexual spores are basidiospores. In Ascomycetes, ascospores. In Phycomycetes, either oospores or zoospores. Here, basidium. Produced exogenously on basidium. On basidium. Whereas ascospores are produced inside the ascus. Fruit bodies, basidiopause. Good examples: Puccinia, Ustilago, Agaricus, Lycoperdon. So this is a rust fungus on wheat, uridial stage, terial stage, teletospores, bicelled, single-celled, binucleate. This is a typical mushroom, my own photograph. Deuteromycetes, the last one. This is again interesting. Commonly called fungi imperfecti or asexual fungi. Generally in fungi, sexual stage is called perfect stage. Asexual stage is called imperfect stage. These fungi always exist in asexual state. There is no sexual reproduction, even if it occurs very rarely. So, they are called fungi imperfecti, asexual state. Saprophytes and parasites. Mycelium branched septate, just like Ascomycetes. Hyphal cells, heterokaryotic, multinucleate. In a cell, there will be many nuclei, but genetically, those nuclei will be different from one another. Hetero, different karyons. Genetically, different types of nuclei exist in the same cell. That is heterokaryosis. Mostly exist in asexual state, produce conidia on conidiophores. Asexual fruit bodies, pycnidium, acerulus, cinematum. Sexual reproduction is partly replaced by parasexual reproduction. Earlier I was referring true sexual reproduction, plasmogamy, karyogamy, meiosis. They occur in a sequence. Plasmogamy will be followed by karyogamy, karyogamy will be followed by meiosis. But in parasexual reproduction, these three stages will be present, no doubt, but they do not occur in a sequence. That is called parasexual. True sexual reproduction rarely form. Examples: Alternaria, Drexlera, Trichoderma, Phoma, Polytota. So these are some of the fungi of Deuteromycetes. Alternaria causes early blight. This is Carolaria. These two are Drexlera. This is acerulus type of fruit body. Sometimes flask-shaped, closed flask-shaped structure, that is will be called as pycnidium. Here sporodochium. This is cinematum. Conidiophores, fruit bodies, asexual fruit bodies. Remember, these are asexual fruit bodies formed by asexual reproductive structures. This is end of fungi. Now here we try to understand what are the characteristics of the fungal kingdom. What are the different modes of nutrition? Saprophytic, parasitic, symbiotic. Classification: Phycomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes, their characteristics and